start. Thank you. So today, this next talk is going to be on mechanism of resistance to ALK inhibitors. So the previous two talks already covered methods of testing and okay, so we go back to mechanism of resistance to ALK inhibitors. So the previous talks are discussed on the various drugs which are used in the first line setting for treating ALK mutation positive non-small cell lung cancer. And sir had also discussed about the resistant mutations. And I'll be discussing in the next 15 minutes a bit about the resistance to ALK inhibitors in lung cancer. So we know, like sir had discussed in the previous talk, that the limitation of first-line or ALK TKIs, including the next generation ALK TKIs, is the development of resistance, which again leads to limits the benefit with the TKI. So there can be two types of resistance. One is primary resistance, which is the less common resistance we encounter in clinical practice. It's actually very rare, where the patient does not get any kind of response within the first 3 months of using a TKI, and it's defined as primary resistance. So there is an intrinsic lack of response to the TKI. And the more common of what we encounter in our clinical practice is usually the acquired secondary resistance. And this is not just for the ALK mutations, it's commonly seen in the most of the driver mutations, including EGFR. So what are the types of ALK resistance? One we saw was the primary and secondary, depending on the initiation of treatment or not. That is, those who receive TKI and do not benefit within 3 months are primary, and secondary are those who progress after some benefit to a TKI. There are other way of looking at the resistance mechanism is on the molecular pathway that they basically have the resistance in. So one are the on target or the ALK dependent mutations, which like the other driver mutations, for example, EGFR, where there are mutations in the tyrosine kinase domain of the ALK protein, fusion protein, and these lead to resistance. And the other types of mutations are the ALK independent mutations, where the ALK protein is not involved and it's not a target mutation. They develop in other pathways. There may be alternative driver mutation pathways that get activated. There might be downstream pathways beyond ALK, which might get activated and again lead to resistance. Some transformations like the epithelial to mesenchymal transition and transformation to small cell lung cancer. So this diagram shows the same. So you can look at the protein, that is the ALK protein seen in blue and green, and then the on-target mutations in this protein due to change of point mutations or other structural changes. And then the alternative pathways, that these are the bypass pathways, get activated. These are the independent mutations, ALK independent mutations, and the downstream pathways that can get activated and lead to mutation. So going to ALK dependent resistance or the on-target resistance. So the main question is, does our prior treatment impact the resistance that we develop? Like we know that patients who are treated with first generation EGFR TKIs typically develop the T790M mutation in 60% of the patients. So does the scenario replicate in ALK positive lung cancer? So it is slightly similar, where resistance mutations after first generation ALK TKIs like crizotinib, they have a particular pattern, and resistance mutations after the newer generation of TKIs are slightly different. So the most common mutation is the gatekeeper mutation, that is L1196M, which is seen in patients who have received prior crizotinib. Basically, it is a mutation in the part of the tyrosine kinase domain of the protein where the ATP binding pocket. Similarly, the next most common mutation which is seen in patients who have progressed on crizotinib is the G1269A mutation. And the other mutations which are seen at these various residues at these positions are seen after progression on crizotinib. Now, this mutation, that is a G1202R, will be discussed a lot. The importance of this mutation is it not only confers resistance to the first generation TKIs, but all the other second generation TKIs are resistant and only sensitive to lorlatinib. So these are the common mutations in the tyrosine kinase domain in patients who have received prior crizotinib. There are some other pathways. One is amplification of the ALK fusion gene. So this is a rarer type of resistant mechanism, but it can be seen in patients who have received prior crizotinib, and again, alternative pathway activation. So approximately 1/3 of patients who have received crizotinib get ALK resistant mutations in the tyrosine kinase domain. And these are the alternative oncogenes, that is the alternative pathways, which is approximately another third. And about 18% get these ALK copy number gains because of the amplification of the gene. So what is the difference again in the pattern of mutations within the first and second generation TKI? So this is from one of the early papers published in 2016, where they had done a comprehensive genomic profiling of patients who progressed after all the various TKIs, crizotinib, ceritinib, alectinib, lorlatinib, and they looked at the type of mutations that were seen. So if you look at the pie charts, they show the proportion of various mutations in different settings. So these are patients who were resistant to crizotinib, these were to ceritinib, and these were to alectinib. And if you look at the pattern, this is all the ALK wild type. This dark blue is ALK amplification, which is one of the pathways for the resistance to crizotinib. And here is the most common mutation that you see with crizotinib resistance, that is the L1196 in the gatekeeper site. But if you look at the higher generation of TKIs, you can see that again, ALK gene amplification is not a common mechanism of resistance. If you look at this part of the pie here, which is maximum in alectinib, that is the G1202R. So looking at this pie chart, we know that the profile of resistance also varies. So the G1202R is seen more frequently in patients who receive ceritinib, brigatinib, or alectinib, that is the second generation ALK TKIs, rather than the first generation crizotinib. So this is the paper which again did the initial comprehensive analysis of all the mutations in biopsy specimens after progression. And like the similar pie chart I showed you, this bar graph also shows that patients who have received a second generation ALK TKI have a much higher frequency of getting this G1202R, and that has a significant impact on our treatment decision, because we know that these patients are not going to respond to the other second generation TKIs, and we need to use lorlatinib. Also, this shows all the various mutations and the frequencies. So this is crizotinib, ceritinib, alectinib, and brigatinib. We can see the most common mutation in the first generation is L1196M, but that is not the case in the second generation drugs, where G1202R is common. It's up from 21% to 43% with brigatinib, as we noted. And again, ALK mutations in general, that is having a mutation in the ALK protein, are in general higher in patients who have received second generation TKIs in the first line than just receiving crizotinib. So this study showed that these ALK resistant mutations were there in 56% of patients overall, and only in 20% of those who had received crizotinib. The spectrum was different, and like I mentioned, G1202R was the main mutation that was seen. And the reason they propose for this mutation being common in second generation ALK inhibitors is because of the binding site. The second generation ALK inhibitors are more effective, and they have it because of the structure of the compound, which binds to the solvent front region on the protein, where this G1202 site is located. And this increases the activity, and that's why these drugs are more effective. But because of this potential of this drug binding, this also induces resistance mutations in this region and makes it difficult to target. So this again is from the same paper, where they did genetic alterations and the various ALK mutations and other non-ALK pathways which were analyzed in patients receiving these drugs. So this shows the efficacy of all the 5 ALK inhibitors in various ALK mutations. The ones, basically it shows the IC50. So if you have a green block, means your IC50 is less than 50 nanomoles per liter, which means your drug is very efficacious. The red indicates resistance, and like you can see, G1202R is resistant to crizotinib, alectinib, and even has a very high IC50 for the other 2 drugs, that is ceritinib and brigatinib, and only lorlatinib has an IC50 of less than 50 nanomoles. So let's come to double and complex mutations, that is having more than one ALK tyrosine kinase domain mutation in a patient. And this is also seen in EGFR. So what does it indicate, or what does it mean if you have a complex mutation in ALK? So these are some reports where they have reported complex mutations, that is at two different sites on the protein. And this usually, if you look at the drugs used, this comes after sequential treatment, after sequencing one ALK TKI after another. And they have tried other TKIs, including crizotinib in this particular ALK mutation, which basically resensitized it to the action of crizotinib. So one thing to remember is that usually these complex mutations might be resistant to the early generation TKIs, but not all complex mutations increase the resistance. So some of them will resensitize. So this was the mutation which is shown in the table as an example. And this was published by Alice Shaw as a case report in NEJM way back in 2016, where they reported a patient who had developed a resistant mutation to crizotinib, that was C1156Y in the tyrosine kinase domain. And it was resistant to ALK. And subsequently, when they developed a second mutation, which made it resistant to lorlatinib, that is L1198F, this mutation basically made it sensitive to crizotinib again, and the patient was shifted back to crizotinib. So this is another paper which has shown the clonal evolution actually of the various mutations over a long period of time, from 0 to 60 months in a patient who received different drugs, erlotinib, chemo, then ALK was identified, crizotinib and brigatinib, and how the ALK mutations progressed over time. They had done a genomic profiling. So the rare mechanism of ALK resistance is epithelial to mesenchymal transition, and I will discuss this slide later. So basically, it is a change in the epithelial cell property. They lose certain molecules like E-cadherin on the surface, and they lose the polarity, and they become more invasive, and they circulate throughout the body, and they migrate and invade. And there are some signaling pathways which are affected in this, and it contributes to resistance. Sometimes you might get the common gatekeeper mutation along with the epithelial to mesenchymal transformation. And how did they identify this? So what did they look at? Pre-crizotinib, H&E staining, and they did a E-cadherin staining, and you can see it is lost here. And some of the mesenchymal IHC markers appear. So this is vimentin, which is a mesenchymal marker, and it was seen after this transformation. Then what about resistance to next generation ALK inhibitors? So these are some papers which have been published. Resistance to lorlatinib. So basically they have seen that a lot of patients obviously who receive lorlatinib do not receive it in the past, did not receive it in the first line setting, and they were received it sequentially. So a lot of mutation resistance was because of complex mutations or multiple mutations, and the detection of more than 2 ALK mutations was significantly higher in patients who relapsed on lorlatinib. So lorlatinib is active against G1202R because that, and it is a primary resistant mutation to the other ALK TKIs. So only lorlatinib is active against this. And also, like I mentioned earlier, that if you do a whole exome sequencing in patients who have received lorlatinib, you can see a stepwise accumulation of the various ALK mutations over various sequential treatments. So how do we detect these ALK mutations? You need to do a comprehensive genomic profiling. Usually ALK mutations are detected using NGS. You can rebiopsy, or including, there have been studies with liquid biopsy. I will not go into detail with it because the methods have already been discussed. But then a question comes is, what happens if we encounter resistance mutations? How do we treat the patient? So this is what we commonly encounter in our clinics, where the newer generation TKIs are cannot be afforded by a lot of patients. And once you relapse on crizotinib, you usually move on to a second generation ALK inhibitor. And if you progress on that, and then we usually prefer, if the patient is fit, to do a biopsy if possible. If there is a resistance mutation, you would offer a third generation ALK TKI depending on the mutation. If there's no resistant mutation, it depends on if any alternative pathway is there, and that can be targeted. If there are no targets, then we have to resort to chemotherapy. So these drugs can be used after the progression of on crizotinib. Now all of these drugs are used in the first line, including lorlatinib has data for use in the first line setting. So in the future, there could be more resistance mechanisms, and the resistance mechanisms post lorlatinib might be complex and might be challenging to manage in the future. So generally, if you have used any of these new drugs, that is the second or third generation TKIs in the first line setting, you would prefer to do a biopsy to identify if there are any ALK resistant mutations, and you can appropriately target them. And with that, if you have a TKI, you see the IC50 and you treat. If you don't have an on-target mutation, you have an off-target bypass pathway, you treat accordingly. And if there are other mechanisms for which there are no actionable or new targetable drugs available, then you go to chemotherapy. So this is just a figure which shows, and just shown in the previous talk, the various ways you can sequence the drugs available and get maximum benefit. So with this, I'll end my talk, and thank you for patient listening.